Welcome to Let's Get to the Marks. Going through GCSE Combined Science Trilogy paper, Biology Paper 1, 2020. It's an hour and 15 minutes and there are 6 questions. I suggest at the start of each question, you grab a bit of paper and a pen, you pause the video, have a go, and then unpause and watch the walkthrough. Let's get into it. A student investigated the effect of different concentrations of sugar solution on pieces of potato. This is the method used. Cut 5 pieces of potato. Record the starting mass of each piece of potato. Place each piece of potato in a different sugar solution. After 24 hours, remove the pieces of potato from the sugar solution, record the final mass of each piece of potato, calculate the change in mass for each piece of potato. Question 1.1. What is the independent variable? The change in mass of the pieces of the potato, the concentration of the sugar solution, the length of time the pieces of potato are in the solution, the starting mass of the pieces of potato. Of course, the independent variable is the variable that you're actually changing, not the experiment. So what is it in this method that you have changed? Place each potato in a different concentration of sugar solution. So you're using a different concentration of sugar solution. And so the answer is this second one here, the concentration of the sugar solution. And that'll get you a mark. All right, let's move on. We have a results table here. Table 1 shows the results. We've got the concentration of the sugar solution. We've got 0, 0.1, 0.2, 0.3, 4, and 5. We've got the starting mass of each piece of potato. Notice they're all slightly different. We've got the mass of the potato after 24 hours in grams. And we've got the change in mass. Now, the first question, explain why the potato in 0 moles of sugar increased in mass. So if we look here, this one that is 0 moles of sugar, so no sugar in the solution, it's just water. The potato went from 7.94 to 10.14, increased by the most mass. It went up by 2.2 grams. So what has gone into the potato and increased the mass? Well, hopefully you've worked it out. Water has, moved into the potato. What's the process by which water moves into things? Osmosis. So water has moved into the potato by osmosis, and that has got us a mark. Do you remember the concentration movement? So water moves from a high concentration. Now I should say a high water concentration outside the potato to a low water concentration inside the potato. And there we go. That'll get you the second mark. Cool. Okay, what we've got here, we've got a little graph. So 1.3, complete figure 1. Some of the results have been plotted for you. You should plot the data from table 1, draw a line of best 3, a line of best what? A line of best fit through all the points, and it's worth 2 marks. So what we've got to do here, we've got to put 3 points on. They've already done 3 for us. Notice there's a negative part and a positive part. So we're doing the changes of mass that were positive. So we come up here, they want you to plot these 3 sets of results on against these 3 concentrations. So we've got 2.2 when it was 0, 1.15 when it was 0.1, and 0.25. So let's go put those on. Now, important when you're doing a graph is to work out how much each square is worth. So the value is 1. And how many squares? There are 10 little squares. So each square is worth 1 divided by 10 is worth 0.1. So each square is worth 0.1. And our first was 2.2, our first result. And that was at 0 concentration. So we want to start on the line, come up to 2.2. And there is our first plot. Now at 0.1, it was 1.15. So that is 1. And then 1.15, well, each one of these is worth 0.1. So it's somewhere between the second square, in the middle of the second square. Now they're not going to mind if you can't get that exact. And then 0.2 was 0.25. So again, each square is worth 0.1. So we're going to go 2 up and then halfway up, third square. Now, to draw a line of best fit here, you might be thinking, do I do a straight line? And I'm just going to show you why you don't. Look, you're going to miss a load of points there. Now, when do you know to use a curve? When there is a natural bend in the results. And it's a nice slight curving like that. And you can just draw a nice straight curve all the way through the results. That is not a nice straight curve. So let's try that again. And that is not a very good curve because I've missed 3 of the results. The key is that you don't take your pen off the paper. What they don't want to see you do is this. They don't want you to take your pen off and do that. They want a straight curve through all the results. So let's try this again. When I say a straight curve, they don't want the curve to be straight, but they want it to be a smooth line. That is rubbish, but that will have to do. Now, question 1.4 says the mass of the piece of potato does not change. Let's read the rest of this question. When the concentration of the solution inside the cells is equal to the concentration of solution outside the cells, determine the concentration of the sugar solution inside the potato cells. So what you have to do here, what we've got to do here is work out exactly, that is not very accurate, that line. Let's get that line back there. Okay, so what they want us to do here is work out where is it that the potato has gone into the sugar solution and after 24 hours it hasn't changed. And that is going to be the place where the amount of water moving into the potato by osmosis is exactly equal to the amount of water moving out by osmosis. And that means that the potato will not change. And you can find that on this graph where the 2 lines intersect. So right here. So that means that right there is the concentration of that potato. And it is 2, it's less than 2.5. I'm going to go 2.4. You might go 2.35. So I'm going to say my answer is it's 0.24. And that's my predicted concentration of the potato. It's wherever the line crosses the concentration line. Okay, let's move on. So once again, my answer for this would be 0.24. Now let's go to the next question. Table 1 is repeated below. Calculate the percentage change in mass for the potato at 0.2. So here we're looking at this column here. And it says use the equation percentage change in mass is equal to change in mass over the mass of the potato at the start times 100. Give your answer to 3 sig fig. So what we're looking at here is we're really looking at the 0.2 row of results. So they want us to look at this. They want the change in mass over the mass of the potato at the start times 100. So what was the change in mass? At 0.2, it was 0.25. So we want to write 0.25. We want to divide that by the mass of the potato at the start, which was 7.96. How do I know this is at the start? Because in the table there it says start. So 7.96, and then we want to times 100, and we are going to get our answer. So 0.25 divided by 7.96 times 100 equals 3.14, And on my calculator, it says 0.7. Now, this will only get you 2 marks, and you'll have to kick yourself if you've missed the third mark, because the third mark is give your answer to 3 significant figures. So I need to only put 3 of these numbers down. And as the 0 is less than 5, we're going to keep it as 3.14. And that gets you the third mark. All right, let's move on. Let's move on. I have a cold today, so if I sound a bit dreary, it's because I've got a cold. How many marks did you get out of 9? All right, don't forget, grab a piece of paper, grab a biro, pause the video at the start of this question, have a go at these, and then unpause and watch me go through. Okay, starch is digested to form sugar molecules in the digestive system. What is the name of the enzyme that digests starch? Well, the enzyme that digests starch is amylase. Now, most enzymes that you have to know at GCSE end with the letters A, S, E. So think about the other enzymes. You've got protease, ends with A, S, E. You've got lipase, ends with A, S, E. So these enzymes, this one does digest starch. Protease is nice and easy. It digests protein, and lipase digests lipids or fats. Where are most food molecules absorbed? Large intestine, liver, small intestine, stomach. So have a go. I'm going to go through the answers. The large intestine is actually responsible for absorbing water, and you need to know that. So if you don't, note it down on a flashcard. What does the liver do? Well, it's got about 500 jobs, but in digestion, the liver produces bile. And the bile is the substance that neutralizes the stomach acid and emulsifies fats. It has 2 jobs. The small intestine is where small food molecules are absorbed. So this is the correct answer. I'll give you an example of some food that's absorbed in the intestines: glucose and amino acids, which you get from breaking down protein. Those are the small food molecules we're talking about. They're small enough to enter the blood. And then the stomach digests protein. Also contains stomach acid to create the correct pH for protein digestion. And so our correct answer here is this one. And there we go. It shows 2 villi. Figure 2 shows one cell on the surface of a villus. That's the singular for one of these. Both of these are called villi or villi, as seen using an electron microscope. So here's the microvilli with mitochondria in it. Give one advantage of using an electron microscope compared with using a light microscope. So the thing about an electron microscope is that you can see small detail like the mitochondria. So we can say you can see more detail. You could say clearer images. I don't know why I've got a W there. Clearer images. But the best answer, it has better resolution, or instead of saying better, higher resolution. And that basically means that you can see more detail or clearer images. You can see smaller organelles, like mitochondria. With the light microscope, you can only really see the cell wall, cell membranes. If you're looking at a plant cell, you can see the cell wall, you can see the nucleus, but you can't see a lot of the smaller organelles like mitochondria or ribosomes. You wouldn't be able to see. Whereas an electron microscope, you can see them. What's the reason? A beam or a ray of light is about 200 nanometers thick, whereas a ray of electrons are much, much smaller than light. So you can distinguish clearly between 2 objects with a ray of electrons, or 2 small objects. Okay, let's go down. What type of blood vessel is labeled X? Tick one box. Is it an artery, a capillary, or a vein? Well, let's take a look. So we've got the food molecules coming across here. So think about it. You've got like, ooh, you've got your food molecules moving across. If they're going to get into the blood and be taken off, there needs to be tiny holes in these blood vessels here, labeled X. So these are capillaries because capillaries have tiny gaps in the cell wall, or not the cell wall, I shouldn't say that, in the capillary wall. There's no cell wall in a capillary, it's made of animal cells. So there's no cell wall, but it has tiny gaps in the walls of the capillary that allow small molecules in and out. So the answer is capillary. Just like in the lungs with the alveoli, it's the capillaries that pick up the oxygen and release the carbon dioxide. Right, 2.5. The real length of one villus is 0.8 millimeter. Calculate the image length if the villus is viewed at a magnification of times 20. So what we're doing here, I like to think of this triangle, I am, and anything that's at the top of a divide line, you can put in the triangle. So I put the image size up there, and then underneath the magnification and the size of the object, real size of the object. I call it the actual size, and I put a times because I am sounds better than Okay, all I need to do, this helps me with rearranging equations. So if I know the real length is 0.8, that's down there, and I know the magnification is 20. So if I want to find the image size, I do 0.8 times 20. So we're going to go 0.8 times 20, and that's going to give you 16. So the answer is 16 millimeters, and that'll pick you up 3 fat marks. Figure 3 shows two cells from the surface of a villus. There are sugar molecules inside and next to each cell. Sugar molecules inside and next to the cell, inside and next to the cell. Cell A, cell B, and the key, the little black dot here, is a sugar molecule. Name the process by which sugar moves into A. So hopefully you can remember the 3 processes. Name the process by which sugar moves into B. And this happens in the first chapter of biology, and it's called cell transport or transport in cells. There are 3 ways that things can move: diffusion, osmosis, or active transport. So those are your choices. Pause the video, have a go, unpause, see if you get it right. Okay, the first one is diffusion. Why? How do we know it's diffusion? It's because it moves from a high concentration, where there's lots, to a low concentration. The second one isn't diffusion, it's moving from a low to high. And so this is active transport. Now, active because it requires energy to move those particles against the concentration gradient. You might ask yourself, but why isn't it diffusion? Isn't diffusion also high to low? Sorry, why isn't it osmosis? Isn't osmosis always high to low? Osmosis is always high to low, but this is talking about sugar, and osmosis is only about water. So anything other than water, if it's going from high to low, diffusion. If it's water, it's osmosis. Anything from low to high, active transport. Give one use of sugar in the body. Well, sugar is a great energy storage molecule, so we could write that. Or it is used in respiration, would be the best answer, to release energy. And that could be energy for growth or energy for contracting muscles, for moving, for lots of different things. Okay, let's look at 2.9. 2.9, figure 2 is repeated below. So we've got this exact same image of 2 villi, or each one of them is called a villus on their own. And then we have these microvilli that are all round the edge. Explain how the villi are adapted for efficient absorption of sugar molecules. Let me move down my screen. Now, this is worth 4 fat marks. Let's get into it. What do you need to say? Well, it says explain. So you're going to describe what a good exchange surface has. Now, this is in the small intestines for absorbing food, but you'll find most of these things are also found in the alveoli or the lungs that give the alveoli the ability to absorb oxygen. So anywhere we're going to absorb stuff in our body has to have three main descriptions, and that is they have to have a large surface area, they have to have a good blood supply, and they have to have thin walls. And this applies to the lungs. This applies to the small intestines. This applies to the gills of a fish. All of these things have those three descriptions. But if you just memorize that, that's just going to get you 2 or 3 marks because the question actually says explain. So we need to say a large surface area because there is microvilli, which are these things here, and they're highly folded. The villus themselves have a large surface area. So I think there's 2 marks there on offer. One for saying large surface area because of the villi, or even larger surface area because of the microvilli, the further extra folding. So because there is microvilli means rapid diffusion. So you're going to get a quick movement of food molecules. Now we have a good blood supply. Why is that important? Think about this. You have food molecules here. When they diffuse over, it's not going to be long. If I have 5 food molecules here and the concentration after a while, we're going to end up like that. And we're going to have an even amount of food molecules. And then the food is going to start diffusing back out. And we don't want that. We want the food to carry on diffusing in at all times. So if you have blood, the blood sweeps away the food molecules like this. So it's always a high concentration outside to inside. So more food moves over, but then the blood comes and sweeps away the food, takes it off around the body, always leaving it high to low. And there's a way we can describe that, which is a lot simpler. We say a good blood supply maintains a steep concentration gradient. So this is a really good line to learn on a revision card. If you're explaining why it's important to have a good blood supply in an exchange surface like the alveoli, fish gills, or the villi in the small intestines, it's because a good blood supply maintains a steep concentration gradient for rapid diffusion. Now, the last one, thin walls. If you had thick walls made of lots of cells, so if these are all the cells of the villus wall, then the food has to diffuse through all of these cells before it can get to the blood. Well, here, if you zoom in there, there's only one line of cells. They're 1 cell thick. So thin walls, you can say 1 cell thick, but what is the purpose? Thin walls have a short diffusion. Now you can say distance, or you can say a short diffusion pathway. It's totally up to you, but short distance for molecules to diffuse. And that is going to get you all 4 marks because you've got 3 descriptions with 3 explanations. So technically, you've kind of said 6 things, so it's going to get you the 4 marks. You really need at least 2 things with the 2 explanations to get the 4 marks. There is an extra one here. If you look, they've really highlighted that there's mitochondria. Now, the exam board aren't just doing that for fun. They're giving you a clue. What is that for? So we'd say lots of mitochondria release energy for active transport. Do you remember we said if you're moving at some point, all the food has moved across and you've got a low amount of food left. How do you make sure that all the food gets into the blood? Active transport can move the food from a low concentration to a high, and that requires energy. So cells that are doing exchange of substances often have lots of mitochondria. Okay, being overweight can affect the health and life expectancy of a person. 3.1, give 1 disease related to being overweight. So pause the video, have a go. Okay, I'm going to go through the answers. So it could be arthritis. That's the one where your joints hurt. If you're overweight, you can put a lot of pressure on your joints. You could get diabetes. Now, the exam board would prefer you to say type 2 because that's the one that is influenced more by our environment. We could also get heart disease, or if you want to be more technical, you could say coronary heart disease. But those are just some of the diseases that you should know off by heart are caused by being obese or overweight. Right, let's look at the next question. 3.2, body mass index helps to show if a person has a healthy body mass for their height. BMI is calculated using the equation BMI equals body mass in kilograms over the height in meters squared. Notice the little square sign. A woman has a BMI of 27 and a body mass of 68.1. Calculate the woman's height in meters. So we know that the BMI is 27 and we know the body mass is 68.1. What I like to do is use the magic triangle. I've already talked about that. This line means divide. And what we're going to do here is body mass is above the line. So I'm going to write my body mass here, which was 68.1. I'm going to put BMI underneath and one more thing underneath, and that is the height in meters squared. So if I want to find out the height, I'm going to do the body mass divided by the BMI, and that's going to give me the height squared. So the body mass is 68.1. I'm going to divide that by 27, and that's going to give me the height squared. Now, what you could do is just find that answer out and then square root the answer, or you can square root the lot. But let's see what it is. Let's do it both ways. So 68.1 divided by 27. This is going to pick you up at least 1 mark, possibly 2, and that is 2.52. So that is going to get you a mark. Now, if you forgot to square root it, you won't get the rest of the mark. So we want to go 2.52 squared like that. And that's going to get you the marks. So square the answer. And you're going to get 1.588. And I'm going to round that up because 8's bigger than 5 to 1.59 meters. Now, in order to get that, I could have just got rid of the square there by square rooting both sides. So you could just do the square root of 6.8 over 27 and smash that in your calculator. Don't forget to put those things in brackets, but you'll get 1.59 and you'll pick up 3 beautiful marks. Okay. Let's go down to 3.3. Figure 4 shows a height body mass chart for adults. So we've got the height and we've got body mass. Notice the height's not squared. It's just the height body mass chart. Which weight category describes the woman in 3.2? Tick one box. Right, so in 3.2, we found out that she was 1.59 meters and she's 68.1. So we go back down with that data and we go 1.59 is about there. Let's move along to 68. So she is bang smack in this gray bar here, and that is overweight. So just to show you how to do that, you could have started at 68.1 and draw a line up, and then you get up to about 1.59. So she is definitely, even if you thought she was 1.6, she's definitely in this region here, and that is overweight. 3.4. People are encouraged to control their body mass with diet and exercise. Describe how the balance between the mass of food eaten and the amount of a person exercises controls body mass. So the mass is equal to Einstein said that mass and energy are the same. So E equals mc squared. Mass and energy are the same. So if you want to keep your mass the same, don't take in more energy than you use up. So we want to say input energy. Oh my gosh. Okay, there we go. I'm ready now. Input energy must equal output energy. So if you are eating a lot of food in diet, it is high energy. You need to use lots of energy when exercising. You need to do lots of exercise to control your body mass. So if you don't want to put on weight, then the amount of exercise you do has to equal what you take in, the energy you take in. If you want to lose weight, you have to do more exercise, use more energy than you're taking in. And then some of the mass on your body will be converted to that energy because you're not eating enough, and you'll lose weight or lose body mass. If you want to gain body mass because you feel like you need some more body mass, then you need to take in more energy from eating than you're releasing from exercise. And that's the balance, the energy balance. It's as simple as that. Okay, 3.5, we've got a big, fat 6 marker. During long periods of vigorous exercise, the body respires anaerobically. They're not telling us that for nothing. It means they want us to talk about anaerobic respiration. And they're telling us that the exercise is vigorous. Explain the changes that happen in the body during and after vigorous exercise. Right. If you're doing vigorous exercise, then you're going to be doing aerobic respiration and anaerobic for that little bit of extra energy. Think about exercise. What are the physical changes that happen while you're exercising? So there's two main ones. We're going to be breathing faster and you're going to breathe deeper as well. Heart rate beating uh, more. You could also say we're breathing faster, but we're also breathing deeper. So when you're exercising, you're going, you're breathing deeper. If it's vigorous, you're breathing that air in really deep. Okay. You're also probably sweating a lot, but this is more of an extras one. So these are three changes. We want to explain those changes. Why do you think you're breathing faster, and why is your heartbeat beating more? So I'll talk about the sweating one after, but we'll put that as an extra at the end. So breathing faster and heart rate beating more. Um, Oxygen is needed. What for? For aerobic respiration to release energy for muscle contraction. So whenever you move your body right now, you're moving your arms if you're writing something down, or whatever part of your body you're moving, you have to squeeze, contract muscles. When you're exercising, you're contracting muscles a lot. So we need oxygen to the cells, the muscle cells, so they can do aerobic respiration to release the energy to squeeze those muscles and to move. So obviously, the more you're exercising, the more you've got to get the oxygen there. And the heart rate increases blood flow to the muscles, and the breathing rate brings in more oxygen to the lungs. So that's our explanation for why that happens during exercise. What about after vigorous exercise? Well, when you're doing vigorous exercise, actually, we're still during vigorous exercise, because oxygen supply, this means how much oxygen you can breathe in, um, cannot meet the demand during vigorous exercise. So when you're exercising hard, you can't get oxygen in quickly enough to do enough aerobic respiration to supply all the energy. This means that we end up doing some anaerobic respiration. And what is anaerobic respiration? Inside your muscles is a store of glucose, and glucose can be turned straight to lactic acid, but it releases a little energy. So, because oxygen supply cannot meet the demand during vigorous exercise, the cells can do anaerobic respiration. What is anaerobic respiration? This turns glucose straight from glucose into lactic acid and a little energy. So essentially, anaerobic respiration produces lactic acid, but it does make a little energy to help you with that vigorous exercise. What's the problem? Lactic acid is toxic and makes muscles tired and painful and sore. It fatigues them. After exercise, our heart rate and breathing rate remain high while we take in more oxygen to remove the lactic acid. The volume of oxygen we take in is called oxygen debt. So this one is a little bit strange, but you've probably noticed when you stop running, you still breathe hard. Now, why would you still breathe hard? You've just finished running for a bus. Why are you still going? You shouldn't be breathing hard because you're no longer moving your muscles. And if you're no longer moving your muscles, you no longer need to do aerobic respiration to release energy to contract those muscles. So why is it that for 60 seconds or so after doing exercise, you're still out of breath? That's because while you were running, you produced some of this toxic substance called lactic acid in your muscles, and it must be broken down and removed. And the amount of oxygen you need to breathe in after exercise is called the oxygen debt. It's how much oxygen your body is owed to get rid of the dangerous lactic acid. Now, I did mention sweating. We could talk about sweating. We sweat in order to keep our temperature stable because respiration as a side effect releases heat energy. So when we're doing aerobic respiration or anaerobic respiration, we're breaking down glucose, they're exothermic reactions and they release heat to the surroundings and that warms our body up. Now, so that our enzymes don't denature, we sweat in order to keep our core body temperature below 40 degrees and at the optimum, which is about 37.5 degrees, we sweat a lot during exercise. And that's the reason why. Okay, let's move on. Question 4. Cells are the basic units of all forms of life. Describe 4 differences between a bacterial cell and a plant cell. So pause the video. Have a go. So here's my lovely jubbly bacterial cell, and yeah, if you don't know how to do this, it's the best way to revise. Grab a bit of paper when you're watching Netflix and scribble down a diagram of a bacterial cell and then try and name everything in the bacterial cell. And so, does a plant cell have a cell wall? Yes, a plant cell does have a cell wall. But a plant cell has some extra bits that a bacteria cell doesn't have. Okay. And you just got to think about the differences. Okay. Didn't really leave space for that big thing. Okay, that's my crummy drawing of a plant cell. There's my bacterial cell. I think the sizes are way out. The plant cell's about 100 times bigger than the bacterial cell, but there we go. Okay, so I could start with that. Plant cells are bigger. So we want to say the difference. Bacteria cell has no nucleus. What does that mean? The DNA is floating in the cytoplasm. So I could say that's a separate point as well. In bacteria, DNA floating in cytoplasm. It's just loose in the cytoplasm. Then what is the shape of the DNA? So in bacteria, DNA is circular. So it's in like a big shoe. Imagine taking a shoelace, tying it together at the end. It's like a shoelace that's been tied together at the end. Of course, in plant cell, you could say the opposite. You could say DNA is a double helix. Just like in ours, we have a double helix with our DNA. Bacteria do not have a double helix. They don't have that twirly whirly DNA that we have. So a plant cell has that inside its nucleus, and the bacterial cell just has this floating circular DNA. And then, of course, the bacterial cell has, so bacteria has plasmids is another one. Plant cells don't have plasmids. The cell wall is different. They both have a cell wall, so that's a commonality, something that's the same. But a bacteria does not have cellulose in cell wall. It's actually made of something called a glycoprotein, peptidoglycan. It's a sugar and a protein joined together is what a bacteria has in their cell wall, but they don't have cellulose in their cell wall. The other thing is the bacteria cell sometimes has a flagellum. So that's another thing we could say. It has a tail called a flagellum. And also the bacteria cell has smaller ribosomes. They're known as 70S ribosomes, whereas the plant cell has 80S ribosomes. So there's lots and lots of differences. Make sure you could get at least 4 of them. And if you don't know them, get them down. But whenever you're doing a question on differences and you're practicing, you're not yet in your GCSE, I would be thinking in my head, okay, if I had to name 3 things that are the same between a bacteria cell and a plant cell, could I do that? And of course, they both have a cell wall. They both contain DNA. Might not be, one might be double helix, one might be circular, but you can just say they both have DNA. I could say they both have ribosomes, they both have cytoplasm, they both have a cell membrane. So look, there's a whole heap of things that are the same. So make sure you watch out. This was specially focused on 4 differences, but you could be asked to say 2 differences, 2 similarities, or to compare and contrast. Right, let's zing. 4.2. Gonorrhea is a bacterial disease. A new vaccine is being developed against gonorrhea. Describe how a vaccine would work to prevent gonorrhea. Now, this doesn't matter whether it's gonorrhea, whether it's herpes, whether it's COVID, whether it's flu. We just need to, for GCSE, know how a vaccine would work. And so this is how a vaccine would work. It would contain dead pathogens. We could say dead gonorrhea pathogens, or dead gonorrhea is a bacteria, so we could say dead bacteria. Now you can say inactive or weakened pathogens. I've just put dead. You could also say it contains, I'm not going to say gonorrhea antigens because I don't want to spell gonorrhea, contains pathogens, antigens. So it could contain the antigens, the protein markers of that pathogen. And that is going to pick us up a big fat mark for describing what a vaccine is. And then white blood cells produce antibodies. So the key when you're doing vaccines, you want to be thinking of dead pathogens. The next keyword is antibodies. The white blood cells then produce antibodies. Now we need to say these are the correct, or you can say specific antibodies that are complementary to the pathogen's antigen. That's the marker. So it's really important that the antibody is the right one for that bacteria. Now we can say the antibodies kill the pathogen. So the antibodies actually kill the pathogen. So my main thing is to say for a vaccine, it contains dead pathogens, they make antibodies, they kill the pathogen. That's the easiest bit to say, right? Dead pathogens, the white blood cells produce antibodies that kill the pathogen. Bosh. It's even better if you can say correct antibodies that kill the pathogen. So you could get all these marks just in one sentence. But here are some extra bits that you should consider. When you want to say the body makes, say the immune system for these questions. The immune system makes memory cells that can produce the correct antibodies. The response, the second time you're infected, actually, it's not really the second time, it's the first time you're infected with the live bacteria, will be larger and faster. So when you have memory cells, you will have a larger and faster response. Something to describe that, just mentioning memory cells, you will have memory cells will get you a mark. And then, of course, how does it prevent gonorrhea? We need to say people who are vaccinated cannot spread the disease as they are immune. This protects the unvaccinated. So you're actually protecting the unvaccinated. Now, you might be like, oh my gosh, that is a long sentence. People who are vaccinated. So if 15 people in your class are vaccinated and 5 other people aren't vaccinated, because those 15 people can't catch it, they're unlikely or less likely for the 5 people who haven't had the vaccine to catch it. So they're being protected by the vaccinated. And we call this herd immunity. And just mentioning herd immunity will most likely get you the mark. So that's the vaccinated people protect the unvaccinated. So you just need to get 4 of these points down. But I'd say the easiest ones for those of you that are like, oh my gosh, it's so much to revise before my exam, just get a revision card out, write vaccines, explain vaccines, and then on the back say they contain dead pathogens which produce the correct antibody to kill the pathogen. And bosh, you're going to get most of the marks. That's a level 7 or 8 answer. Another disease caused by bacteria is salmonella food poisoning. In the UK, chickens are vaccinated against salmonella bacteria to reduce the number of cases of food poisoning in humans. Explain how vaccinating chickens reduces the number of cases of salmonella food poisoning. Well, we've just kind of gone a little bit through that. If the chickens, whoopsie, cannot catch salmonella, then the humans are less likely to catch salmonella. So it's less likely to spread to humans. So once the chickens cannot catch salmonella because the chickens are immune, so yeah, it doesn't spread to the humans. We get the idea, Tom. No need to waffle on with that one. Okay, let's move down. Give one way the spread of salmonella food poisoning from one human to another is controlled. Do not refer to vaccination. So let's just say you've been unlucky enough to eat some uncooked chicken and you've caught salmonella, or you've been washing some chicken and you forgot to wash your hands and you've touched your mouth and you've had some raw chicken juice in your mouth and you've got salmonella. How can you stop other people in the house getting it? So there's a whole bunch of things. So good hygiene. That means washing hands, washing surfaces. It doesn't spread by coughing salmonella. So washing hands, washing surfaces, isolating, that kind of thing will be good. So like washing hands and washing surfaces is important. Good hygiene. The number of cases of salmonella is usually higher in the summer than in the winter. Suggests why? Well, in the summer, microorganisms have more kinetic energy for their enzymes to work, which means that all living things reproduce faster and better in the summer. So we're going to say they reproduce faster. Now, they do accept on the mark scheme more barbecues, so more uncooked meat, because people notoriously, when they cook barbecues, cooking chicken on the barbecue is a risk because often the outside of the meat gets burnt by the flames of the barbecue and appears to be cooked, but is still bloody or uncooked inside. So that is when we get the biggest spread of salmonella. So there's more uncooked meat going around, basically, is another potential answer. Okay, question 5. This question is about photosynthesis and food production. 5.1. How can oxygen production be used to show the rate of photosynthesis? So you've probably seen this. Oh my gosh, that's terrible, isn't it? Sometimes they put like some pondweed inside a flask and it releases bubbles of oxygen underwater, and we can count the bubbles over time. So count the bubbles per minute. Now, do you need to say per minute? No, you do need to mention time. Whenever you see the word rate, make sure you mention time because it's basically speed. So it's something divided by time. So count the bubbles per minute. We can also use something called a gas syringe. Now, a gas syringe is a much better way of calculating the rate because with a gas syringe, you're going to be able to measure the volume. And the problem with bubbles is they're all different sizes. So we could measure the volume of oxygen over time. What do I mean by over time? I mean divided by time. So measure the volume of oxygen in 10 minutes. Measure the volume of oxygen every 30 seconds. As long as you mention time, you're going to get the marks.
Right, we're looking at this graph. Scientists investigated factors affecting the rate of photosynthesis in tomato plants. The tomato plants were growing in a commercial greenhouse in the UK during winter. Figure 5 shows the results. So let's zing down, peeps. We've got the rate of oxygen here. We've got different conditions here. You can see this line's 30 degrees, that's 20, that's 25. We've got different concentrations of carbon dioxide, 0.1, 0.1, 0.04, that's less than half those. And then the percentage of carbon dioxide in the Earth's atmosphere is 0.04, so that's just normal air. The light intensity in lux, we've got 10,000, 20,000, 30,000, and 40,000 going across the bottom on the x-axis, and we've got the rate of oxygen production given off. Name the factor that's limiting photosynthesis at point X. So on any photosynthesis graph, and this one is about photosynthesis because oxygen being given out, on any graph of photosynthesis rate, whatever is along the bottom here is limiting when the line is going up. So any point here, light is limiting on this line, light is limiting on this line, light is limiting on this line. Light is no longer limiting here. Why? As I increase the light from 30,000 to 40,000, there's no more oxygen being made. That means the plant is not respiring any faster. So I'm giving it more light, but it's not doing anything. That means light is not limiting. It doesn't need light. Whereas here, if I go from 500 or 5,000 to 10,000, look, there's a big increase in the amount of oxygen. It means the plant was saying, give me oxygen, give me oxygen. So wherever the line is going up, the thing on the x-axis is the limiting factor. So the limiting factor at point X is light intensity. It's important you know how to read these light intensity graphs, and if you practice all the past papers before your GCSE exam, you'll have had a go at a few of these questions and you'll be totally fine and prepared. So make sure you do all the past papers. Farmers growing tomatoes commercially try to control the rate of photosynthesis and make maximum profit. A farmer can control the temperature and the carbon dioxide in a greenhouse. What what is the minimum light intensity a farmer should use to get the maximum rate of photosynthesis? Now, they said the farmer can control the temperature and the CO2. So obviously they're going to give them 30 degrees, the best temperature, and they're going to give them zero point 0.1 CO2, the highest amount of CO2 they can give them. But what light intensity should they use? Should they use 40,000 or 30,000 or 20,000? Well, if we have a look, they definitely don't want to use 10,000 or 5,000. If they use 5,000, well, they could give them more light and get a faster rate of photosynthesis. But once you get to 20,000, there's no point in giving them 30,000. Look, it's made no difference at all. And there's no point in giving them 40,000 because presumably using 40,000 is twice as expensive as using 20,000 light, and you get no increase in product. So we're going to say 20,000 is the light intensity they should use because it's the point at which the graph straightens and where light is no longer a limiting factor. Light intensity, the light intensity you gave in 5.3 may not give the farmer maximum profit. So they're now saying that even though we come to this graph, even though we look at this graph and we give them the best light for growing the plant the fastest was 20,000, but they're saying actually it might not give them the most money. And here is a kind of difficult question. We have to consider that maybe the cost of producing that light, the electricity that we need to produce that amount of light, 20,000, is worth more than the increase in plants we get. Maybe it's better to use 10,000 and you get a high amount of oxygen. So you get a high production of your plants. They're growing fast and it doesn't cost a lot. Whereas when we use 20,000, maybe the increase doesn't justify uh, the cost. And in fact, if you look here, look, if I drop down to 15,000, it's going to be a lot cheaper, 25% cheaper, I presume, because it's a quarter less, 15,000 light. And look, it's hardly any difference in the production of photosynthesis. So there's not going to be much difference in the size of my plants, and yet I've saved a quarter the cost. So a farmer has to weigh up costing as well. So let's put that into our answer. Um, they have to consider cost. Um, electricity stroke energy costs money and is used to make the light intensity. Other light intensities are cheaper and still give a good rate of photosynthesis, like 15,000 lux. There you go. I'll give him my own little example to show the examiner that I've been reading the graph and trying to answer it well. And then explain the results when the light intensity was 0. So let's go take a look at the graph and see if we can explain those results. So when there's no light, we'd think, well, there's no photosynthesis, so there's no oxygen, right? Yeah, that's right. There's no oxygen at 0. Oh my gosh, there's negative oxygen. How can there be less oxygen than how much is produced from 0? So my calculator produces 0 oxygen. How can these plants be producing a negative amount of oxygen? That means they're taking in oxygen from the atmosphere. Why would they be taking in oxygen? What is it that, even though there's no light and they can't photosynthesize, so they can't produce oxygen, what is it they're doing that all living things need to do to stay alive? Why are they taking in oxygen from the atmosphere? Why have we got a minus oxygen? So, of course, you should know the answer is they're doing aerobic respiration. So, what we want to say here is there is no light, so the plant cannot photosynthesize and produce oxygen. So there's a couple of reasons why this is getting you the marks. The examiner finds out that you know that light is needed for photosynthesis. So they go, good, they've tested you on that. Is light needed for photosynthesis? Yes. And they've tested you to know that photosynthesis produces oxygen. So lots of marks there. There's no light. The plant can't photosynthesize, therefore it can't produce oxygen. There is negative oxygen production because the plant is still doing aerobic respiration, which uses oxygen from the atmosphere. And that is also probably worth 2 marks. The fact that we're saying the oxygen was negative because the plant is doing aerobic respiration and it's taking oxygen in from the atmosphere. So you need kind of 4 of those points. They like to test this. They tested this in the last GCSE. They tested this in 2023 with a weird question with trees and kind of like showing them in the daytime, then at night, and gases going in and out. Some weird messed up question that was. Okay, but they like to test the fact that plants also respire and the effect this has on photosynthesis and respiration. And there is actually a point where the rate of oxygen production from photosynthesis is equal to the rate of oxygen absorption from respiration. We have that point as well to consider sometimes in questions. Let's get on to the last question. Okay, new drugs tested and trialed before they can be licensed to treat patients. Figure 6 shows how much time the different stages of testing took for one new drug. So we've got the preclinical testing. That looks like it took 3.5 years. Then we've got phase 1, looks like it's about a year. Phase 2 looks like about a year and a half. And then phase 3 looks like 1, 2, 3, 4 years. So you kind of got 4 years, year and a half, 5.5 years, 6.5 years, the whole of the clinical phase. And then we've got the review, the data, and approve takes 2 years. So the whole from starting to test a drug to when it's on the shops, in the chemist, in Boots and other pharmacies is 12 years. So how much more time did clinical trials take compared to preclinical testing? How much more time? Well, it was 6.5 years we worked out for the clinical trials, and the preclinical trials were 1, 2, 3.5. So 3.5 to 6.5. You do the maths, 3.5, and this was 6.5 for the clinical trials. So the difference between them is 3. During phase 1 clinical trials, low doses of the drug are tested on healthy volunteers. So just one reason why we use low doses. So why only use a little amount of drug? Well, in the first stage of testing, the first time people have had it, we want to make sure we use a low dose in case there's side effects. So in case there are side effects, if it makes people's hair fall out, we want to use a low dose so that not too much of their hair falls out while we look for the side effects. Remember, the phase 1 of the clinical trials is the first time they've tested it on humans. 6.3 suggests 2 reasons why healthy human volunteers are used in phase 1 clinical trials. Why do we use healthy people and not sick people with the disease that we're trying to cure? Well, healthy people, less chance of the side effects being dangerous. Like, let's say the sick people have high blood pressure or something, and this one of the side effects is it increases blood pressure. Your drug could actually kill the sick people because one of the side effects is raising blood pressure. So there's less chance of side effects being dangerous. And there's another way we can word that, less chance of the side effects adding to the sick people already have symptoms. This is another point. Sick people already have symptoms. It could be hard to tell whether the side effects are from symptoms. So if you take someone who's completely healthy and they don't get headaches and they don't get dizzy and they don't get nausea, that if they start reporting headaches, sickness, and nausea, it's the drug. Whereas if you take a sick person and you test the drug on them, they might start complaining, oh, I get these headaches. But you normally get headaches anyway because of your illness. And then you go, oh, is it the drug that's causing the headaches or is it because they're already sick? So it's better to test it on healthy people. That way you don't get confused about the side effects, whether it's symptoms of the disease or whether it's because of the drugs. Now, let's look at 6.4. The results of the clinical trials can only be published after peer review by other scientists. Suggests one reason why results must be peer reviewed. So the revision card standard answer is to avoid bias, but also to check the results. So there's also to check the evidence, So there's all sorts of things that you can check. If you say I've got the cure for cancer, maybe you just want to make a load of money and you're kind of like lying about your results. Other people need to check, are your results true? Can they repeat? Is your experiment repeatable? So that's why other scientists will do it to see if it's repeatable and they can get the same results. And that avoids any bias. People are influenced by money and other agendas. Okay, and this looks like finally, a drug is only licensed for medical conditions it was tested to treat in the clinical trials. Drug regulations control what a doctor can prescribe. So we have these things called drug regulations. So doctors can't just give you any drug. Ensure doctors can only prescribe a drug with confidence and they protect patients. The regulations do. AMD is an eye condition that can result in very poor vision. Doctors treat 40,000 new cases of AMD each year. 2 drugs licensed to treat AMD in the UK are drug A and B. In many other countries, drug C is used to treat AMD. Drug C is only licensed in the UK to treat cancer. The cost per injection of each drug is £561 for A, £800 for B, and only £28 for C. Why don't they use C? It's so much cheaper. The number of injections required to treat AMD is the same for each drug. Wow. In 2018, the High Court in the UK gave permission for drug C to be used to treat AMD. Evaluate the decision to allow drug C to treat AMD in the UK. So at first, it looks like it was very sensible to allow drug C to be used to treat AMD. But some people say it was never tested for anything other than cancer. So it hasn't been tested for AMD. So this would be against it. So against, Okay, so generally when I see an evaluate question, I want to do 3 points for, 3 points against, and then give a seventh point, which is my final opinion. To be honest, you probably only need 2 for, 4 against, or you can do 3-3 or 4-4, 2 against. And one of those can be your final opinion. So here am I against. This drug C has not been tested for AMD in the UK. It might not be as good as treating AMD. It might not be as good at treating the AMD as drugs A and B. So that's 1 point. And then you probably get a separate point for saying it's not been tested in the UK. We don't know if there's side effects. It was definitely told us that it was against UK regulations. Now, that's not a law, but it's still breaking the UK medical regulations, which are recommended. But 4, it'll save money for the NHS. Any calculation to show that, i.e., if you do the cost of A and B divided by the cost of C, you can find out how many times cheaper it is. You can treat more people for the same amount of money. It's being used in other countries, so that means those countries have probably tested it. And then I would finally end with my opinion. I'd say I agree with the decision. Because it is used in other countries, it's probably safe. That says sued. It's not been sued in other countries. It's been used. That's just my spelling. In other countries. So therefore, it's probably safe. And that will get you all 6 marks. Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.